Learn how to predict functions of phage genes. This video is going to be divided up into different parts that address location and synteny, looking at consensus with BLAST-P, structure with HH-Pred and deep TM-HMM, and finally how to load things into your Pecan notes and your Benchling notebook entry. So the steps to calling a gene function involve first looking at the location of the gene in the genome for indicators of function, and we'll mostly use Famarator for this. Determining what the consensus is in the community on function for the gene fam. We'll use BLAST-P for this, but we'll also go to the PhagesDB page for the gene fam and look at the list of functions assigned to that gene. We'll predict the structure of the protein and compare it with known protein crystal structures using HH-Pred. We'll identify transmembrane domains using the program DeepTM-HMM. And finally, I'll show you how to enter all this information in your Benchling electronic notebook and in your Pecan notes. Let's begin looking at the location of the gene because this can really inform our function calls. And we can do this from a bird's eye view, just looking at our general model for a phage genome where we know structural and assembly genes are on the left side of the genome, and genes that have to do with DNA metabolism or gene regulation, etc., are found on the right side of the genome. And in the middle, we find the lysis cassette, the integration cassette, and the immunity cassette. So this could be important, for example, if we're working on the left side of the genome, we would expect to find structural genes, right? But if we're working on the right side of the genome and we come across some data that points towards, for an example, a terminase, our alarm bells would go off and say, well, gee, that doesn't make sense for this area of the genome. So just knowing what to expect in that region of the genome can help us make good decisions about the individual genes that we're analyzing. But I also want to point to the fact that the organization of the genome for prophages is a little bit different. And since we have some class members that are analyzing prophages, I wanted to remind you that the genes get shuffled around a little bit once a phage genome integrates into a bacterial genome. So let's take a look at how that works. We learned in the first semester that a phage genome has an attachment site at P that allows integration into a site in the bacterial genome at the at B site. These two sequences are identical or nearly identical, and the phage integrase will make a cut in the at P and a cut in the at B, and the phage genome goes in, becomes a prophage. That prophage genome is defined now by at L and at R, which are just hybrid versions of at P and at B. But once that integration occurs, because the cut happens at the at P site, what's to the left of the at P will be one end of the genome, and what's to the right of at P will be on the right end of the genome. And so just looking at this model of an integrated genome right here, we can see that red represents the integrase, followed by the immunity cassette. Then the replication genes, the COS sequence would be in the middle, and the replication genes would be on the right. So let's look at that. Here we have a prophase genome, and in this version of the prophase genome, again defined by at L and at R, I actually have the integrase on the right side of the genome, adjacent to at R, but the integrase can be on either side. It could be immediately adjacent to the at L. It depends on where the at P site is relative to the integrase. So sometimes @P comes after the integrase and sometimes it comes before. That will impact which side of the genome you find the integrase. But other than that, usually what you see is the immunity genes are usually on the left side. And so this might be an immunity repressor. And then divergently transcribed from that is the first replication gene, which is CRO, followed by the rest of the replication genes, the COS sequence, the structural genes, the lysis cassette, and then something that I call accessory genes. These are genes that are usually located between the integrase and the lysis cassette, and they're typically expressed during lysogeny and have the potential to impact the fitness of the bacterial cell. So if you're working with a prophase genome, take a minute to draw out a model of a prophase genome so that you know whether you should be working on replication genes or structural genes or whatever. We can get more granular about location by thinking about synteny. This is a screenshot right from your bioinformatics guide that reports the order of structural and assembly genes as they are found in most cyphovirus genomes. And so you guys can open up your guide and have this list. But basically, this allows us to use synteny as part of our evidence for making gene calls. And I'll use the example of the gene that I'm going to be calling today. I'm looking at a gene in the Clipper genome that is going to be preceded by a terminase and followed by a capsid maturation protease. And so I'm going to predict that that gene is likely a portal protein because that is the syntenic location for a portal protein. But we can also use synteny in other parts of the genome, maybe not for the entire right arm, but often genes occur in groups if they are all related to the same biological function. So for example, would be adjacent to the integrase cassette is the immunity cassette. We know that immunity repressors are usually encoded in the reverse direction, and that divergently transcribed from the immunity repressor is the CRO gene. So if we found the immunity repressor, the next thing I would be looking for is a gene going in the forward direction that has helix, turn helix, DNA binding domains, and is a potential CRO gene. There are also lots of other examples of gene pairs. I'm just going to give you one more. For example, many of you have genomes that have toxins. And so most of the time, phages that encode toxins also need to encode the antitoxin so they don't die from their own toxins. So if you find a toxin, you'll want to look at the gene either to the left or to the right of the toxin to see if you can find an antitoxin. Let's look at GP9 in the Clipper genome. This is the gene that I'm continuing to call from our previous video. I've already called the translational start. And I've aligned the Clipper genome with MatCat and MEGSI. We can see that there's high nucleotide identity between the genomes in this region. We can see that this gene is present in MatCat and in MEGSI, and they have both assigned a portal protein function. Also, if you look above the gene, the number is the FAM number for that gene FAM. And then in parentheses are the number of members in that family. So this is a really big family with over 2,000 members. The other thing that I can look at is in MatCat, which has all the functions present, the gene upstream is a terminase and the gene downstream is a capsid maturation protease. That gives me a pretty good idea that this is likely a portal protein based on function calls in other genomes and also its location between a terminase and a capsid maturation protease. So let me show you how I entered some of this information in my BenchLink notebook. So this is a continuation of my notebook entry for Clipper GP number 9. Here's my screenshot. I've circled the gene that I'm interested in. And I've written a little bit about the synteny. So, for example, I've noted that the gene is downstream from a terminase and upstream from a capsid maturation protease, and it's the syntenic location for the portal protein, and that MacCat and Megzi have called this gene a portal protein. I did a little comparative genomics too. So I noted the FAM number and that this is a really big FAM with over 2,000 numbers. I also did a little bit of investigation in Famerator and found that this FAM is found in many, many different types of phages from different clusters. So I listed just a few that are mycobacteriophage clusters, but also noticed that there were clusters that include phages isolated on different bacterial hosts from different genera, including Gordonia and Curtobacterium. So how did I figure that out? Here's my live Clipper map. And I think because new genomes have been added in to the PhagesDB database, this FAM has actually changed since I took that last screenshot. So notice I have a new FAM number, and now this says that there's 2338 members. So just within the last week, the number of members of this family have grown, and that's because the Hatfield lab is continually adding newly sequenced genomes. But this is the same gene, that's gene number 9, and has the stop codon 4599. If I click on it, I can get information about the DNA sequence, the protein sequence. I can learn whether there's any conserved domains, which we'll talk about later when we look at structure, if there's membrane domains, but I can also get a list of the phages that contain this gene FAM. And so look at all these different clusters. The A's, it seems to be in every single subcluster of A, the AZs, it just goes on. And down here are the K's, and Clipper is a K1. You can click on the cluster, and then it gives you specific names within that cluster that have that FAM. And to me, it looks like probably every single cluster K genome has this gene FAM. So are there any ones in here, like including Banana Dare Elf and including Lava Hound, which are some of the Ks that we're calling in class this year. So I have noted that as well in my comparative genomics. And then there's a place here to write other map observations. This is where you want to write whether there's been insertions or deletions in your gene relative to the genomes that you've aligned, because those could be some important observations that help you make decisions about your function. So that's all I have right now for location. Hopefully this will get you started thinking about why it's important to think where your gene is in the genome as you begin to gather your thoughts about the potential function. In our next video, we are going to talk about consensus. What does the rest of the community think the function for that gene is?